So we had our data set for about 8 hours. We were working on it. We would analyze, produce our figures, and put it onto a publicly accessible website. 8 hours later, the US group would take over, and they would start posting the analyses that they had done overnight and their interpretations, and keep going. And it cycled over and over.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. You may be familiar with open source software, but how about an open source pandemic?
Our guest today is Dr. Justin Bahl, faculty member in the Department of Infectious Disease and the Department of Epidemiology and Biostatistics, as well as the Institute of Bioinformatics. Welcome to the podcast, Justin.
Thanks for having me.
So our favorite first question is about your pathway to science. When you were 2 years old, did you know down to your core that you wanted to be a scientist, or what was your pathway?
I think I always knew. I dabbled in trying to be a professional musician for a little bit, but deep down, I always wanted to be a scientist.
That's cool. In high school, did you have opportunities to go along that pathway, or was it not till college?
I toured the U.S. performing when I was in high school.
Oh, did you? Okay.
From the science side, I just enjoyed the science classes, and I enjoyed learning. It was really into university that I learned what aspects of science that I enjoyed the most.
What type of experiences did you have then to kind of get you on the path where you are today? Or maybe they didn't.
Well, I can tell you that my first couple years of university almost turned me away from science. I was very excited to go to university and to be in this learning environment, but the classes were massive and impersonal. I went to the University of Toronto. Our intro bio class had 1,700 students.
Wow.
Holy cow.
So it was huge. Maybe you had a lab, you got to interact with a graduate student that was running that particular section, but it was very difficult. And the first 2 years, it really was about learning where you wanted to be. And I wasn't sure I wanted to be in university at that point. That didn't seem like the place that was best for me. Third year university, though, I started getting very interested in botany and plant pathology, and the classes became much smaller. I got to interact with professors, and I got to ask questions until they said, nobody knows the answer to that. And that was amazing to me. My third year university, I realized that I could go out into my backyard and discover a fungus that nobody has ever seen before. And I would be the one that gave that to the world. And that was where I became excited about studying science and doing research.
Cool. And so at some point you decided that you had to go on to grad school.
My journey into science is very convoluted.
Oh, we want to hear about it. These are the ones we love to hear about.
Yeah. So I was very interested in mycology. It was the most fascinating set of organisms that I encountered at that point because they had an asexual form and a sexual form that were morphologically so different that for a long time, people just didn't know they were the same organism. And trying to resolve their evolutionary relationships was a question I wanted to pursue, which meant asking very general questions. I explored grad studies in the US, and I would have been pigeonholed into working on one particular plant pathogen if I came to the US. Things were becoming very, very specialized early on, and that would have been my option. That wasn't so exciting to me because I wanted to be able to go into the forest and say, what's here, and learn why it's there. So I had an opportunity to go to the University of Hong Kong. And at the University of Hong Kong, I worked on saprophytes that were found on dead and decaying palm leaves. Palm leaves grow in nice places. I did my PhD in a lot of nice places. So I collected samples from North Australia, Queensland, and areas around that province. Collections in Hong Kong, where I was, in Thailand, and in South China. And it was great because I got to ask very general questions like, why is this particular fungus on that palm tree? And then I would look all around in plants nearby and I wouldn't find that fungus again. But if I found another palm tree a few kilometers away, it had that same fungus. How did it get there? How did it move around? Was it always associated with that plant from seed to death? It was a very interesting exploration of biology. At that point, I didn't know that I was actually working on epidemiological questions, fundamental epidemiological questions.
Okay, and I guess at that point you hadn't really taken the step toward molecular characterization yet?
So I was actually. It was a combination of classical morphological comparisons and genetic sequencing.
Okay.
So the genetic sequencing methodologies that we have were first generation in that lab. We didn't even have Sanger sequencing at that point. We had gel plates, and each nucleotide went into its own separate lane, and to prepare the plate took about 8 hours of cleaning.
Banging the plate.
I remember those days.
Yeah. So that wasn't so fun, but comparative genetic methods were growing at that point, and I really just became interested in how to build a phylogenetic tree. What was the best way to make a decision, or not a decision to, well, partially how to best design your data set, how to best do the statistical inferences. And that was a point where new maximum likelihood and Bayesian approaches were becoming dominant. So I got to explore this different area of statistics. Ironically, the two worst classes that I ever studied in university were genetics and statistics. And now I think you could argue that I'm a statistical geneticist, or that's a major component of my work.
So I think we're going to get pretty deep into epidemiology, but before we get there, because the way that you just talked about it, I just think that maybe a general audience listening to this might not understand kind of the breadth of epidemiology. So I think when we talk about when the common people would think of epidemiology as just diseases transmitting, right? But it's much bigger than that. Can you just kind of give us a simple discussion to kind of put people on the same page of kind of what epidemiology is?
Sure. So I start my lectures in epidemiology with actually the etymology of the word. So epi means upon, demos means people, and logos means the study. So it's the study of what is upon people. That can be infectious diseases, that can be chronic diseases, that can be maybe a symptom from malnourishment that's not necessarily one of these other more common thought of diseases. And it's expanded to exploration of social aspects. It's expanded to economics. It's very, very broad in general. But if you think about just anything, if you have an infectious disease study, you might want to include how people actually move and interact. What about the proportion of school-age children that are being impacted, or the school-age children that may be driving the spread of the disease? So you have to explore much more than just that actual instance. There's this idea that it's separate from other aspects of infectious diseases. If we just go specifically infectious diseases, right? Like we have these ideas emerging from germ theory and Koch's postulates, and then we come in and we say, well, we have clinical infectious diseases, and then we have population scale infectious diseases being epidemiology, and we treat them as if they're separate. But you really do need to link them. And I think that separation is very artificial.
Well, we separate them into different departments, really.
Different campuses here.
Yeah.
Kind of makes sense to me now that this is kind of the field you're in based on what you told us about how you wanted to study science and do different things, right? It was kind of this bigger, right, broader look at something versus getting pigeonholed down into something. So it makes sense to me.
So before we go any farther, one of the things on your website that you say you are is a molecular epidemiologist. So let's just get into what the molecular addition to that term means. So
I use genetic data, and primarily I use genetic data from pathogens. RNA viruses, I think I've done one study on a DNA virus and a couple studies on bacteria. Mostly I'm working on RNA viruses, especially those negative sense RNA viruses like influenza. And I compare those genetic sequences to other sequences that are in the public database and try to learn about why we see patterns in its evolutionary relationships amongst those pathogens. The patterns emerge very quickly in RNA viruses because they spread very fast, they replicate very fast, and they accumulate mutations very fast. Those things come together that the signal of geographic spread is observable in the genomes. The signals of host changes is observable in the genomes, and sometimes the signal of social interactions, like spreading amongst children, or particular venue affiliation, or maybe if it's a hospital-acquired infection, the movement between floors even. So if they're accumulating so quickly, we can use an evolutionary analysis of the viral genome to make inferences on the epidemiology that may be determining the patterns of spread.
So how much has all of this next generation sequencing and kind of this molecular approach changed the way that epidemiology is done these days? It's a big question.
It is a big question. And I wouldn't want to say that it's, I would say that the traditional approaches and these new molecular approaches are complementary, and that the most powerful inferences that you're going to make from using genetic data will take advantage of the traditional epidemiological boots on the ground investigation. Any sort of analysis of genetic data has very limited value without the metadata. The metadata in this case would be the date the virus was sampled or taken from its host, the location, and then if you have other factors about the host that might be important, like for example, what species it was, whether it was a juvenile, whether it was a child, or if it was maybe within a long-term care home, or the different context. So when you add all those things together, you get to make stronger inferences, and things make more sense from analyzing genetic data. When you go further into like contact tracing and understanding some of those social aspects, then you can start making inferences even deeper, like maybe resource allocation and how that might be determining pathogen spread. Maybe it's for avian influenza. Maybe it's because there's a water body that they all breed at, and we see that juvenile birds are coming up at staggered time or at the same time, and that's causing an explosion of these pathogens as well. So when you combine traditional epi and molecular epi, it's great. And I would actually say that molecular epi is a subset of traditional epi.
So in the intro, we referred to a open source outbreak, and I think in my notes I have that down as the 2009 H1N1 influenza pandemic. And so what do you mean by open source outbreak?
I would say that everything before that, the data was very difficult to generate that data, and sharing that data was very slow. In 2009, the first individuals that were detected through our surveillance program in California, I believe, they had an influenza that was different than the ones that were circulating. And it was typed and determined that it was the swine origin influenza. So I think it was towards the end of March in 2009. They identified it, and then shortly afterwards, other individuals with the same virus were detected in Canada and Mexico. That raised a lot of alarms. The global community of scientists is made aware of this new event that was happening on, I think, April 24th, 25th. The genome sequence was available the next day, and it was shared with everybody at that point. And now people are collecting samples. They're looking for it. Viruses are being sequenced, and they were being immediately deposited into the public domain. From our side, I was based in Hong Kong. At that point, I had moved away from studying fungi, and I had joined the State Key Lab for Emerging Infectious Diseases, where we specialized in influenza. We were an high-path avian influenza reference laboratory for the WHO. And we had been collecting viruses from throughout Southeast Asia and southern China, Hong Kong. I say we, but my primary role was in analyzing the genomic data. There was an army of individuals working on this. People had been collecting viruses that were circulating in swine. They were going to these abattoirs, going to other sources and collecting samples. And then they had collected a bank of influenza from swine. At that point, they started sequencing it. So almost immediately, we're having viral sequences being collected around the world, deposited into a public access domain. Our animal sequences were coming up, and we were adding them into the analyses. And all of this analysis, when we produced a new analysis, we would share it immediately with all the other scientists. So we knew within maybe 2 days that the swine origin pandemic had emerged from a swine population, but the gene segments had some of them ultimately came from avian sources, other ones came from human sources. They had mixed together in the swine population, and we were sharing this information in as near real time as we could. We partnered with a group in the UK, two really important researchers in molecular epidemiology, Andrew Rambo and Ollie Pybus. Andrew at University of Edinburgh, and Ollie was at Oxford at that time, as well as another individual, or a few individuals, but in particular one in Arizona, Mike Roby. So we had our data set for about 8 hours. We were working on it. We would analyze, produce our figures, and put it onto a publicly accessible website that Andrew Rambo had hosted. So we posted up there, and other scientists would comment on the interpretation, suggestions on changing the analysis, and everybody else could observe this. Then we would essentially hand off our data set. Somebody else would start producing their data set. We would clean up our results based on comments, produce a nice figure, and then the other group would lead the analysis, and we would start commenting on their work. It was a very exciting time. We would get 2 or 3 hours of sleep, but
it's very real time.
It was very real time. And then 8 hours later, the US group would take over, and they would start posting the analyses that they had done overnight and their interpretations, and keep going. And it cycled over and over. And colleagues at that point said to me that they weren't going to the WHO for information. They weren't going to CDC. They were coming to our website and just refreshing, saying what's happening. Because we were providing, the best information that we could, and the best data that was coming out at that time was genomic data.
Right.
So that was the first time that things were happening in this framework where you could either participate as a member of science, or really anybody could take that result and make their own comment on it. It was curated, the website, that you had to be a contributor to make those comments, and we didn't just want it to become very noisy, but anybody could take that and make comments on it and do work on it if they wanted to. But it was a near real time and accessible analysis, and we said what sequences we were using. And it was very exciting.
Sounds exciting.
It sounds very exciting. I mean, it's such an anomaly for science in some ways, right? We're so siloed in doing our own thing in our own labs and not wanting to share because we want to get the credit or we want to get, whatever. But especially in epidemiology, you can't work that way, right? If you want to intervene or make some changes or stop something, right, you have to do this in real time.
Yep, you have to. And context is so important, right? And context comes from this broader information. I think if you're going to do interdisciplinary science, you have to acknowledge that you can't be an expert in everything.
Exactly.
You have to learn the language of these other fields, but you cannot actually sit down and say, well, I'm now the expert in epidemiology. I'm now the expert in genetic sequencing. I'm now the expert in evolutionary analysis. I'm now the expert in veterinary diseases or anything else. It's impossible. So you need to learn how to communicate across domains and accept that there are other people that were going to constantly teach you.
And I also like the idea, you kind of briefly mentioned it, is you would also talk about how exactly you were doing something so that you could kind of all do it the same way, right? Because a lot of times also we have some groups that are doing this experiment, but they're using this particular method, which can give you different results from this other particular method, right? So if you kind of coordinate and make it more uniform, then you can really do the kind of cross-study analysis.
And you can examine the limitations of these methods more effectively, and you can have new methods come up. Certainly, so if we go forward from 2009, I think at that point, that philosophy has underlie every epidemic or disease outbreak investigation since, from Zika, Ebola, Mpox, MERS, SARS-CoV-2, the sharing of data and the sharing of analyses in as near real time as possible. People are very open with it. And what we saw during SARS-CoV-2, first, I think I saw at one point over 4,000 papers being published a week. It was intense, and everybody in the world was working on this. Within our domain, we saw an explosion of new methods and new approaches to analyzing the genomic data. We have new tools for visualizing data that make it more accessible to people. I saw people had created a little website, and the next day it was in the New York Times, and people were exploring it and using it. So I think the rate of method development, the rate of data sharing, and the rate of sharing of results to make them accessible has also increased in this field. So I think I point to 2009 as the moment of where open source epidemic investigations have started, but it's exploding right now.
It really is.
So one thing I want to touch on a little bit. So of course you work with molecular data, mostly RNA viruses, but you've also mentioned the importance of metadata. And maybe you can tell us, in a perfect world, what kind of metadata do you have with your, tagged along with your molecular data?
Mm-hmm. I think the absolute minimum is the exact date of isolation. I would love to have GPS coordinates as to where it actually came out. I think city is great. If we only have state, we lose some power. If we only have country, when we're looking at really big countries or really small countries, the power of inference, like we can't treat them all equal, but we have to, because that's the limitation of the data. And that's great for fast evolving pathogens. I think we run into some ethical walls very quickly when we talk about pathogens that evolve slower. So, if we have the exact date of isolation and city for a pathogen like pertussis, if a child dies, we might be able to identify exactly who that child is from that news report and the actual pathogen. So that becomes one of those things that we have to be aware of, that we can't have that same metadata for everything.
Okay.
So something like SARS-CoV-2, providing city information and the exact date of sampling had very little risk to individuals because it was spreading so fast and such high numbers were being infected that there was no way that you would be able to say, oh, that was you. You got us sick, right?
Right.
And we don't want to start criminalizing individuals for saying, I just went grocery shopping.
Right.
Yeah.
Not knowing. Right.
Yeah. So there are variations in that metadata. But I think that's the minimum for fast evolving pathogens. I think if we can start including things like age, even more data like zip code, for example, of where that person resided, age, school district. These are things that we can use, but we have to take careful steps to ensure that we are protecting patient information.
So basically, ethical use of data, is that a good way to describe this? Or
It is, and it's a massive challenge when we're talking about academic research versus public health practice. So the state public health lab may have a lot of data that we can use, but we can't just access it. It's being collected under a certain set of regulatory information, and sharing that information even with the next state can be challenging. We're in the state of Georgia. We work a lot with the Georgia Department of Public Health. We have to be very conscious about how we are accessing data and ensure that a lot of the regulatory framework is actually in place. Now, how do you put those things in place for something that hasn't happened yet?
Yeah.
Like if we're talking about like a pandemic, that's really challenging. And putting the proper precautions in is tough. So there are 5 centers in the US that are trying to resolve this right now. And I'm not sure if we've come to a place. I think a lot of us thought that this would be like our deliverable in year 1. Now, as we're coming into year 5, we're still we haven't converged on a solution to that problem in how to share data, even just amongst the centers that have agreed to work together. And within a center, amongst institutions, these are multi-institutional centers, there'll be like a public health lab and academic partners, and we still don't know how to share data amongst ourselves. Now, if you put it on the global scale, it's even more challenging.
So you kind of alluded to how things are really, really hard before a pandemic even starts, right? Because you don't know when it's going to start. So what about kind of the on the ground? Like, are hospitals and physicians kind of trained with like a workflow or something of collecting samples? Like, if they see something unusual.
So once the pandemic starts, people are trained very quickly. Right. They learn that thing. So far, we've discovered pandemic viruses because a very tenacious individual was curious.
Yeah.
That's really what it comes down to.
Yeah.
There was an individual that was just like, I don't know why these kids are sick. It looks like influenza, but it's negative in our influenza tests.
Right.
Oh, we discover swine origin pandemic in 2009. An individual in the Middle East is sick. And they're like, we don't know what's going on here. It's not anything that we know of. Let's collect those samples, find somebody who can try and do it. It gets sent to the Netherlands. We discover a new virus called MERS, right? I think for a large part, that's how it's happened. Things that regularly spill over, like Ebola, which has spilled over many times, people are in those areas are very aware. They're like, I know what this looks like. We need to put protocols in place. But that's a practice response, right? I think we need to train more veterinary scientists that are working in clinical diseases and nurture curiosity for a lot of individuals, even in the medical field as well.
Yeah, that's always so tough when I think about that. I mean, I'm not in that field, but I think about how we have, with training in medicine and different things, it's all about, protocols and maybe not being the physician scientists that we used to have way back when, who were much more curious, right? And that kind of becomes concerning in the idea of pandemics and infectious diseases. But hopefully we just always have that curious person around.
I think the team of people that are working in pandemic emergence are incredibly
curious. I agree with that.
And the clinicians, we're in this point in our lives where there's a new emerging disease. It seems to be recognized very, very frequently. And I think partially that's because we've become better at recognizing these things. And the other part being that we've changed the world that we live in.
That is true.
And reduced the suitable environments for animals. They're coming in closer contact with the animals that we're producing. We're producing more animals than ever. We have armies of individuals around the world that are taking care of these animals to feed our desire for this high quality protein. And it seems that they're spilling over a little bit more.
Yeah, and that's
frequently.
a good point. We haven't really talked about it is, the idea of most of these things are coming from the animal reservoir, right? So we can't really put everything on a physician in a hospital because we should be thinking about it just as much or maybe not even more in the veterinary world.
Yeah, the pre-pandemic preparedness.
Right.
There is one more, and I don't want to get too deep into the weeds here, but you did mention RNA viruses, and we know that RNA viruses have a higher mutation rate than DNA viruses. Is that a good thing or a bad thing as far as molecular epidemiology goes? Because you could imagine that you're having so many changes that it's happening so fast, it's hard to track them, or the fact that there are changes gives you the data you need to separate things. And
so I think both of those are true. You end up with a lot of noise in your data. But if you have an idea about how fast they're evolving and mutations only occur when an individual is infected. So if you're observing X number of mutations, you have a pretty good idea or a pretty good guesstimate of how many people were infected in that chain between two observations. That actually becomes very interesting in trying to use genetic data to try and work out what is the actual transmission chain. We're not there yet, but I wouldn't be surprised if we increase our rate of sampling, go straight to sequencing of samples instead of doing other diagnostic steps where we're doing diagnosis by sequencing. That data is being shared, and then we're able to start producing actual transmission chains. This is not data tomorrow or information tomorrow. It's going to take a number of years to get there. But I think we're going to be able to take advantage of that noise and also be able to deconvolute it to effectively make inferences that will allow us to stop disease transmission. I feel like I need to do more podcasts. I don't get to just talk like this at home.
This is fun. It's a really fun time, and that's why I enjoy it, because it's just so conversational. Okay, Justin, so when you're not tracking worldwide disease outbreaks, what are you doing in your free time?
I have two small twins, twin boys, and they are enormously fun. I would say up until age 4, it was brutal, but they're now almost 7. And
what do they have you doing these days? What's of their interest that you're really enjoying?
They love being out in nature. It takes a while to get them away from the screen. I admit that with 2 kids, give them a little bit of screen time and be able to do the other things that we need to do. But when we get them away and get them into the forest, get them out for a hike, I would take them to go and collect edible mushrooms.
Oh, fun.
They get so excited about this, and now they're collecting every mushroom and they want to know about it. My wife is a geologist, so they're collecting rocks and they're learning about different minerals, and they get really, really excited. They love nature for both of them. And it means that we get to be outside with our dogs and in the forest. And there's a lot of nice places that we can go.
Do you have a favorite place you like to go to around Athens?
The kids really love Lake Herrick. The hikes are not too hard because they are still small, but we can get far away enough or into the forest where we're not running into so many people, and they could find a lot of things to explore, like streams. Streams and their new shoes. They like those, right? Daily hike that I love is Ben Burton, because I can just take the dogs. Kids have been dropped off, take the dogs, and I have like a nice 45-minute hike. And very quickly it feels like if there's no wind or the wind's going a certain direction, you don't even hear the cars. So that's quite nice. And both of those places have a lot of interesting biodiversity. I've found so many box turtles.
Really?
I have found musk turtles, terrapins, snapping turtles, and box turtles in different areas that have gone hiking. But box turtles, just being out in the forest, I thought it was a cartoon that the bottom shell hinges and closes up. But you pick this thing up and you can't actually, it's entirely closed. So they're probably my favorite turtles, and we see them a lot in Ben Burton.
What do your dogs think of the turtles?
They are very good at finding them. They help to find them, especially our previous dog. He would just be able to find turtles. It just made it easier for me to find these other ones and learn about them. The other ones, they're like, oh, this is interesting. Then it closes up the shell. It's like a rock. And they're no longer interested.
Our guest today has been Dr. Justin Bahl, faculty member in the Department of Infectious Disease and the Department of Epidemiology and Biostatistics, as well as the Institute of Bioinformatics. Justin, thanks for being with us.
Thanks for having me. This has been really fun.
Links to additional information about Dr. Bahl's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemeth, for research, editing, scheduling, and keeping us on track.